Yeah. So hello, everybody. What I wanted to talk about this afternoon, so basically before I leave tonight, is I've given it a bit of a bizarre title, and I guess that title is not what you had on your schedule. Sort of applied evolutionary biology, and how it's basically what I'm trying to communicate is how we can use ideas and sort of methods to analyze sequence data to track, predict, and possibly help fight various diseases, right? And most of what this is going to be about is influenza, but I'll try to, I'll talk a little bit about our efforts to broaden the scope to other, mostly viral diseases towards the end of the talk. So how would sequence and evolution help you to track disease? So if you have an outbreak of sorts, and you have infected individuals, which are here sort of these red dots, right? Sort of one individual infects another and infects another, and these are then sort of cured infections. And you have this sort of networks of a tree of infections that's spreading through the population, right? From some of these individuals, you might actually get a sample, and that sample sort of shows up in your biobank or in your sort of clinical laboratory or something like that, and you obtain a sequence, right? So the blue individuals are the ones that are observed. All of the red would be not observed, and that's sort of an important thing to realize, right? That between any two observations of a disease you make, there are typically many unobserved states, right? So they aren't there. But in the process of being transmitted, that virus or bacterium or whatever, it sort of mutates, right? It does it not, maybe because it has a particular good reason to mutate, but many of these mutations that we'll encounter are just going to be, random accidents that happen because the molecular machinery that does the replication is quite error-prone, right? So we have a few mutations here, and that mutation, for example, would group these two things together. That mutation basically separates these two guys from whatever happened later, right? So these mutations that happen at some point in the transmission tree, they record you, at least to some degree, the history of that transmission, right? And we can straighten things out a little bit, right? And then you recognize this kind of phylogenetic tree where sort of different mutations pin together different individuals, right? And the resolution of this is basically limited by the number of mutations that you see or the length of the genome you sequence or something like that, right? Meaning if you want to have a fine-scale picture of the spread of a particular virus, it pays a lot more to sequence the entire genome in fewer people than to sequence sort of a tiny little bit in many people, right? So that's sort of because the information gain, adding more leaves here is typically only logarithmic, while adding more sequence is linear, right? So all these slides are on the web. But you're happy to take pictures. You're welcome to take pictures too. All right. So the one disease where this is kind of most established and probably at present also most useful is influenza. And as you probably all know, influenza, the first big pandemic that is well known, was the 1918 Spanish flu, right? After the First World War, on the order of 50 million people died of the flu in sort of, in a pandemic that spread across the globe within a year or so. We still have samples from people that happened to die in a place that never thaws. There are these sort of permafrost samples of influenza in 1918, which is why we actually know that sequence, right? And so after this huge pandemic, this influenza lineage, this virus, then became sort of a seasonal epidemic that comes back every single winter, and I'll talk a little bit more about that later. But yeah, so there was this huge spike in 1918, and next year is going to be the centenary of this, so expect a lot of flu-themed conferences. That was the H1N1 strain, yes. But that at that point doesn't really mean much because
I may be absolutely off the mark, but I thought H1N1 is a very recent strain.
Well, it's an existing strain, and there's another one here. And okay, hold on 30 seconds. So, H1N1 here really is at first, in the beginning it didn't have a name because there's only one, right? 1957 is when it then suddenly needed to have a name because there was another influenza strain which appeared out of the blue. That is avian birds in this case, which kind of terminated the circulation of this strain. And this new strain, which is then called H2N2, was serologically different, right? So you could tell these strains apart by taking antibodies from people's blood and sort of having them react with the virus. And there were people that were infected with this strain didn't recognize that, and vice versa. And this recognition is actually dependent on two different surface molecules: the hemagglutinin molecule H and the neuraminidase N. And that's sort of how this nomenclature H1N1 and H2N2 emerged. And we now have on the order of 20 for each of them. Because there's a huge diversity of these influenza strains that circulate mostly among birds and waterfowl in particular. Yeah.
Just a confusion, right? So this H1N1 is also evolving.
Right.
So when you say it becomes H2N2...
It doesn't.
Okay. So what's the difference? Yeah.
So these are very different. These are, order hundreds of thousands of years apart, right? So these
but in terms of like protein...
We have no idea for practical math, right? But
as a protein, how different... I mean, you say antibodies don't attach, but how should I think? How big is it?
Align them at the amino acid level.
Right. You can't.
You can. But it's a pretty messed up alignment.
Okay.
while sort of, from here to here, it's sort of very gradual, right? Yeah, so but what happened in 57 is that there's a new strain that no human on Earth, or very few, maybe a handful, had seen at that point, which rapidly spread across the globe and terminated circulation of this one. This didn't have a very long run. It only circulated until 68, when the H3N2 lineage popped up, again sort of out of avian sources, and it took over the N2 segment from the H2N2, right? So influenza you can reassort, so you can recombine these different bits and pieces, but it had a new hemagglutinin, and this is the so-called Hong Kong flu, which has then sort of kept circulating ever since. And this is the main object of what we'll talk about over the next half an hour or so, right? Sort of H3N2 is the most persistent and most virulent lineage these times. So blue means sort of, putative circulation in avians, in birds.
So where the blue arrow connects to 1968, does it mean it hopped from humans to from avian to humans?
Yes.
Oh.
Yeah. So there was a bird that had the virus, and then there was a human that had the same virus, and that happens. So these animal-human transmissions actually happen pretty often in the context of trading of live chicken, farming. Humans give their viruses to the animals too. So you'd be surprised how many human viruses are found in pigs, which is actually what happened here probably. So that virus was all over the planet. Probably at this point made it into pigs, and has been circulating in pigs ever since, until in a very complicated reassortment event in 2009, it spilled back into humans. And because this was such a crazy pandemic back then, people were very, very scared when this happened. And you'll see in a minute why. But the other effect that this had is that this lineage has stopped circulating, so we haven't seen this virus in humans since. And there's another curious bit here. This virus hasn't been in circulation for about 20-ish years, between the late 50s and the late 70s. And when it reappeared in a fashion which was completely unchanged, despite the virus having a pretty high mutation rate, which probably, the virus wouldn't do that all by itself unless it was frozen. And the only places where viruses are consistently being frozen is laboratories. So there is no official version as to what happened here. and since this is recorded, I'm not going to give any specific opinions. Yeah.
One more question. So why is it so episodic? I mean, we have been living with pigs during, go back, right? So this red lineage, right, of H1N1. So why was it sort of hiding for 70 years? I mean, it's not that, so why is it so episodic that, it sort of came.
You're asking, so why are cross-species transmissions so difficult?
So, or so episodic, yeah. In the sense, why it's sort of, we are handling pigs all the time? Is it some special events where the transmission happens, or?
It certainly required, at least, the thing that got transmitted was a quite spectacular reassortment that sort of combined things from many different lineages. So it was a triple hit. So it's not something that happens every day. So we do have sort of, very frequent transmissions of the H7N9 lineage to humans in sort of context of Chinese, sort of, poultry farming in China. But that doesn't tend to spread. So I think the answer to your question would be that it actually happens quite often, that you have some infection, but it doesn't manage to take off, right? But coming back to the seasonality, so if you now zoom into the last couple of years here, sort of what this plot shows you is over the last 20-ish years, the number of positive tests done by the US CDC, so in North America, and the results. So whether that was sort of an H1A virus, an H3 virus, a new 2009 virus, so that's the swine flu, right? It also is an H1N1, but it sort of has here a different label. It's distinguished, or a subtype, or an influenza B virus. I'll only talk about A viruses, but the B viruses say, in some aspects they're similar, in some aspects they're somewhat different. But what you see is this pronounced seasonality, right? So in the Northern Hemisphere, you have, a very well-defined flu season, which typically starts round about now and lasts for 2-3 months and sort of dies down end of February. And some, H3 sort of has been dominating most years. In some years, it's not dominating, right? There's some sort of a competition between the different influenza A lineages. And in 2009, certainly something out of ordinary happened, right? So this is not your usual seasonality pattern. And that is when this new lineage, so that is when this happened, right? So you suddenly had, even the flu season was kind of over. This is zoomed in here. The flu season was over, and suddenly it picked up again. So this spike in the other ones is probably just suddenly heavily increased sampling and testing. So this is not properly normalized for sampling effort. But you suddenly had this variant take off in March, May, April, which was very unusual. And that's what made people so, and knowing that this is a relative of the 1918 flu, that's what made people so scared, right? But it fortunately turned out sort of less serious than it could have been, and has since also settled in this kind of seasonal pattern. Yes.
Sequencing, is it not a relatively recent thing? I mean, how do you know the lineage back then?
We have reasonably good sequencing up until the early 90s. And these tests you could also do earlier based on antibody, right? So you would do serology instead of sequencing, and you would tell these broad categories you can tell apart by very traditional methods that people developed in the 50s.
But the previous slide that you showed, I mean, this one. So there it starts in 1910, right? I mean, are you doing some kind of
1918?
18,
so
whatever.
There's rather little. This is basically inferred. That if it happened here, it probably happened there, right? We have sequences in this area are very sporadic, and there wasn't any degree of subtyping that because there was the only subtype around, right? So there was certainly influenza diagnosis and statistics on that, yeah, but there wasn't that degree of molecular typing, of course. Yeah, so 2014 was a year that was dominated by H1N1. And actually, this year is kind of interesting because we've had 2 successive H3 seasons now, and it's looked like the current season, at least in the US, is an H3 season again. And in Europe, it's a bit of a mixed picture. And it'll be interesting how it plays out because it actually does make a difference in terms of the vaccine. The vaccine is pretty good for the H1 and it's kind of mediocre for the H3. So if it's the case for getting the vaccine is much better if you have an H1 season. It's a good idea either way. But all right. Yeah. So if you now actually, so this is let's go back 2 slides. So this one is the one that had sort of the longest continual circulation, right? And it also is the one that dominates, the red one here, that dominates most years. And if you now look into molecularly what's actually happening, if you do a phylogenetic tree of influenza A, H3N2, segment HA, so this is the hemagglutinin segment that codes for the protein that sticks out of the virus. If you make a phylogeny of that, and you color it by date, so this is 85, right, and this is 30 years later, what you see is that you have this sort of very, very long stretched phylogeny, which in any given year has sort of fairly low diversity, right? Sort of a particular color who is only present, is sort of restricted to some kind of blob like this. But the distance along this tree is quite enormous. There's this corresponds to probably 10% of all amino acid of the HA changed. Come to that in a second. So what happens along this tree along this time is that the virus sort of accumulates amino acid substitutions on the hemagglutinin protein, which is a trimer that sticks out of the virus, and that's what the virus uses to enter cells. And at the same time, it's a primary target for neutralizing antibodies that humans generate against influenza, right? And that's why all these mutations, they kind of pile up here at the tip, because that's where all the antibodies are targeted, and that's where the virus needs to mutate in order to reinfect the same person again, right? And so this is just saying it again. So these viruses evolve to avoid and escape immunity that is building up in this human population, right? And sort of by the virus changing, so less people get immune against it, are immune against the changed variant, then it can sort of reinfect those people, start as a cycle. As a corollary, the vaccines that we get against seasonal influenza virus, they need frequent updating. And each of these crosses here is a virus that was chosen by the WHO to be represented in the seasonal influenza vaccine. And you see that, I don't have the data from these guys, is not included, but basically every 2-3 years, they sort of move that virus forward a little bit, right? And they do that because you have to maintain a good match between the virus population and the vaccine. Otherwise, the vaccine wouldn't elicit antibodies that actually recognize the circulating virus. Question?
Yeah. Can you please comment on the nature of this tree? Because it seems to be like a cumulative cascade rather than a spider-like tree.
Yeah. So this is, most of the other trees that you've looked at over the past 3 weeks were sequences that were taken from one time point, right? And in that case, there was all sorts of different things that sort of coalesced to one common ancestor, right? So here you have sequences taken from many, many, many different time points. And the statement here is that sequences taken from one particular time point, they always have a common ancestor fairly recently in the past, right? And that the population as a whole is turning over. So if you take influenza viruses today, the common ancestor of all these H3 influenza viruses has lived about 5 years ago. And that's exceptionally long. Normally it's more like 2 to 3 years. It's testament of the population being rapidly turning over, meaning being under sort of extreme competition to innovate, basically change things up here, to be able to reinfect people, right? Because staying in the same place doesn't get you anywhere. Let's look at it. This is just sort of a trajectory of one particular amino acid site. And it's kind of instructive to look at. I thought we were already done with that. But this is position 189, which sits somewhere up here, tracked over the past 30 years. And you see that it has changed the identity of the amino acid 4 times over this period, right? It started out being a lysine, and then it changed to an arginine, and that change took about a year. It wasn't quite happy with that arginine, right? It never went to 100%. It kind of wiggled around here. But then it replaced it with a serine, and the serine was just at 100% throughout for about 10 years. About 10 years. And then it changed to an asparagine, and then back to a lysine. Right? And this happens at about, several dozen positions. So it's a very rapid, dynamic turnover of things. And yes, so that's what I wanted to show you. So you've just seen this tree, right? And it certainly has a very dynamic temporal component. And together with Trevor Bedford, who is a scientist at the Freight Hutch in Seattle, we basically thought, it's kind of ridiculous that we are doing these trees by hand every year. Let's do them automatic every week. And that's what this is. You had a question? Has that been answered? Do you still want to ask it?
If we go back to the previous slide, please. So did it start with an amino acid and then came back to the same amino acid?
Yeah. So it's similar to the HIV thing that I told you about last week, where we don't know what the favorite sort of preferred state here is, but there is certainly a sense in which it has sort of a limited repertoire that it can use at every particular site. And some of these might actually be driven to escape, and some of it might just be, it's no longer useful to have that amino acid. Let's go back to what works better.
Right. And it certainly...
We don't know in this context.
Interesting because it doesn't match with the idea that coming back to the same genotype is impossible.
Well, no. The genome that segment is 1,701 amino acid nucleotides long. Coming back to the same place at one site has nothing to do with coming back to the same place as a genotype as a whole. There was another.
As the virus is interacting with the human system, is it possible for the virus to acquire maybe extra genetic material from the host?
Nothing is strictly impossible, but I don't think it has been observed. Why would the virus do that? I don't think.
I'm seeing this as a kind of, trying to invade. Maybe it can acquire anything, maybe extra genetic material from the host, which may not necessarily reflect in the phylogenetic tree.
Yeah, so not that I know of. Yeah, but these are sort of very singular events that happened. It's nothing that happens in the typical coevolution between a virus and its host. Oh no, this is just because there's more. The horizontal, vertical axis in this case doesn't really mean anything. So the evolutionary distance is horizontal. Vertical distance is basically just stacking the sequences on top. And if you have more sequences per unit time, you get this kind of it's a sampling issue. So we've already subsampled this, but here it's kind of the sequence availability is below our subsampling threshold, hence it's sort of flatter.
I had a small question. So is there variation between geographies? And is this data only US strains?
Right, look at the screen. So this is an influenza phylogeny that includes data from the last 3 years. And is currently here colored by broad geographic region. So the blue are the samples from Africa. These would be samples from Europe, China. We also have South Asia here. So that is a little bit different. We can just restrict it to only things from South Asia. Right, those would be the samples from South Asia. And basically what you're supposed to see here, while there is some granularity, these viruses are all over the planet, right? And every major clade is represented in every major geographic region. And South Asia is unfortunately one of the regions that we have sampled sort of rather poor sampling from. So most of these samples come from Bangladesh, some from Nepal. India is very underrepresented, even though it makes up probably for a substantial share of all global flu infections. But the Bangladeshis, they send a lot of samples to the US CDC, and they sequence them and process them. And it would really be fantastic if there was more data from India. But currently, this is really a bit of a blind spot in influenza surveillance. But if you look at say North America, right, it's just all over the place. So there's really very, very little, and I think it's even more pronounced for Europe. There's very little geographic structure in influenza H3 that is. In the other influenza lineage, there is somewhat more. Not, it's still, pretty cosmopolitan, but you see that, pretty much every major area here is represented on every part of the planet.
My question is actually motivated by, is the influenza strain evolving to account for variability, like a human genome will vary slightly between India and the US. So the influenza virus adapts.
Yeah, so that is kind of an attractive hypothesis, and that is certainly true for HIV, in that people have shown that HIV is evolving to the local distribution of HLA types in the population. But influenza has a mostly antibody-mediated immune response, and pretty much every one of us, no matter what our sort of VDJ makeup is, can make an antibody that binds just about anything. And what really is to be taken home from influenza is that the immune response is just very stereotypic against the head of the hemagglutinin, and every human on Earth can do that. And does it in pretty much the same way, and hence there is this possibility to globally escape. Right? A T-cell immunity you can't globally escape because there's a lot of variations among humans. But generally, humans are not that different. So it's very rare that you actually find mutations that are only in one particular region. It's mostly a difference in degree. That there's, this is 40% of people over here have it and only 20% over here. Yeah, so this is this thing that, I think it got updated last week, but you can play around in various ways. Sort of, for example, load a longer time frame. It's about a megabyte or so. So this would be a 12-year time frame. You can, down here you can see different mutations. So here, this would be one mutation which actually came and went again, right? It's sort of position 311 in HA1. And that was a Q for a while, for the longest time, and then sort of turned into a histidine. It's still partly a histidine, right? But there has been a pocket again that reverted back to what it used to be. You can then also, zoom into this and sort of annotate the tree by all the mutations that happened. This here down here will tell you the frequencies of the different mutations and clades in the tree. Right, this is the frequency, relative frequencies of these mutations. So that this one here is one which is quite variable over time. 144 is a very, very important antigenic mutation. So you see it does all sorts of crap over time, right? And because of all this variability, we need to update the vaccines, right? And this tool is this is the current vaccine, which unfortunately is not going to be in the Northern Hemisphere vaccine that we are getting now, but it's going to be in the Southern Hemisphere vaccine for 2018. It would be good if it was already in our current vaccine, but came too late. But the real challenge for influenza is whether we we can sort of go beyond this. This tracking is already good, right? Because you know what's out there. But sort of can we do better and actually anticipate what's going to happen next year, say, or in 6 months, maybe. 6 months would be great already, right? And why is that so important? So what you see in this graph here is a sketch of the schedule on which these influenza vaccines are made. So there's 2 meetings, and this is just for one for the Northern Hemisphere. So there is a strain selection meeting, which takes place at some point in February, where sort of a whole bunch of people from different parts of the globe, there's different WHO collaborating centers, get together and decide whether the influenza vaccine needs an update. Right? And once they have decided what is supposed to be in the influenza vaccine, then sort of the companies will sort of make reassortant virus, those that grow well in eggs, right? Because the influenza vaccine is still mainly produced in fertilized chicken eggs, meaning you grow that. So the chicken eggs, the cells in there, in the fertilized eggs, they have the property that influenza grows rather well in them. Only that it sometimes actually changes while growing in the eggs, and then you have a messed up vaccine, and that's not terribly good. That's kind of what's happening this year. So then it takes a few months to actually make it and test it, and starting September, October, the vaccine is being rolled out, right? But from here to here, it's something short of a year. And you've seen that these transitions from one amino acid to the next, right? That's about the timescale they take, right? So if you are unlucky, the decision that you made here is sort of outdated by the time the vaccine comes, right? And for the Southern Hemisphere, there's another such meeting. There's a whole cycle shifted by 6 months for the Southern Hemisphere. India probably doesn't have a very clear seasonality because it's, more of a tropical climate than sort of a temperate climate. But you then sometimes tend to get 2 seasons rather than 1 season. They overlap, I think, with monsoon season. So the tropical zones are a little bit kind of in between, and there have been discussions of whether there is supposed to be a separate tropical vaccine. But I don't think that has been decided yet. All right, so can we predict, right? So at some level, we have this diverse population, right? So we have many sequences, and they differ at yes.
Year type situation, right? So if you could do good prediction, the lag would still be there, or I missed that.
Well, yeah. So they would, it would still. The prediction doesn't change the time scale of vaccine manufacturing, right? Correct. But you'd hope that you could manufacture something that sort of anticipates the future, that you kind of can, be proactive about the changes rather than react, right? That's sort of the main point, right? So the prevailing strategy, and I don't blame them for that, in the past has been, you stick to what you got until it no longer works, and then you change, right? Because, who knows it's basically, are you more willing to take an audacious decision and be wrong in the end? Or do you basically blame it on the virus, right? So it's easier to blame it on the virus that the virus changed than blame it on you that you anticipated something that didn't happen, right? So, but last on Monday, I guess it was, I told you about how we've been modeling a scenario like this, right, where you have your population that's constantly turning over, it's evolving in the face of an adapting human immune system. There's always many mutations that segregate at the same time, so you think you have some distribution where it's not going to be smooth, right? It's going to be kind of bumpy, but anyway, let's just see what you can do. This part is a population by definition, right? It's tautology. This part is a fit part, will grow, and those will go away. And sort of predicting will mean you want to identify things here at the very tip, right? Only that nobody actually tells you where they fall, right? So the only thing you got is the sequence, and that initially is just alphabet soup, right? So it's not there's no defined map genotype to fitness. And this is this kind of an inverse question is what various people over the past years have tried to address. There's sort of one well-known recent work by Marta Duksha and Michael Lessig, who actually sort of tried to make, and succeeded, I guess, make an influenza-specific model of how do we make an educated guess of the fitness of a particular virus corresponding to a sequence based on what we know about past patterns of circulation and the protein biophysics of influenza. Yes?
Please go to the previous slide. What do you mean by similar size mutations?
Oh, yeah. So this is, it's basically just a statement that if there's many and they make comparable contributions to fitness, you get, by virtue of the law of large numbers, something that's vaguely Gaussian. If you have many mutations, but some of them make huge contributions and others very tiny ones, you don't get a well-behaved distribution. Okay. And it's basically a leftover from a graph that also had a different option.
Okay. So basically, it's a number of genes. The threshold number of genes. Above the threshold number of genes, you say it's like.
Yeah. So how many? How many dice do you need to roll to have a roughly Gaussian outcome, right?
Okay.
I think. In reality, it's probably going to be something between 5 and 8, right? It'd be better if it would be, the match between theory and reality would be better if there was more mutations that made smaller contributions. But just sort of as a starting point, it's a useful thing to do, right? All right, but let's just go through this quickly. So this is a top-down, head-on view of the influenza H1, in this case, influenza hemagglutinin. And what is colored here are sort of different regions of the protein that are known to interact with antibodies. And based on knowledge like this and historical patterns of antigenic change, Marta and Michael sort of came up with an idea that you classify the protein into regions at which epitope mutations occur. So these are mutations that change the antigenic properties and hence are likely to signal an adapted sort of a virus that moved forward, right? And then there's non-epitope mutations, the kind of gray balls. These typically tend to mess with the protein structure. So if a particular sequence has many of those, one could reason that it's probably not going to do that well because it's melting too early or something like that. And then they had sort of another somewhat mysterious component in that model, which they call sort of nonlinear component. But it's basically, and I'll provide rationalization for this this in a little while. It's basically measuring sort of a phylogenetic contribution of growth, and that was sort of the approach that we took. And we only later realized that this is actually the joint at the hip. Only that we didn't include anything else. But what they then do out of these three components, they construct a fitness a score that parameterize the frequency change of a particular clade or particular variant of the flu in season T+1 relative to season T, right? And there's, on the order of 3-4 parameters that are being fit on the past 20 years of data. And then you sort of start extrapolating this in the future. And this is a graph from their paper, 2014, where they plot their prediction against the post hoc validation. And it's certainly, sometimes you get it wrong, but that's what you should be expecting, right? But on average, or in the majority of the cases, if it's on the diagonal, right, that means you made a meaningful prediction. Or at least if it's in this quadrant or that quadrant, it's you at least got the sign right. And the majority of clades is certainly much better than randomly picking, right? Yes, sort of we had a different approach, and that is sort of based on the trees. And before I get into this, I just wanted to, basically formalize the problem a little bit and give you, not the entire mathematical background, but sort of one sort of central little piece that I hope is not too scary, but it's sort of an instructive little calculation. So can we? Nobody there? I'll figure it out myself, even if I'm not supposed to. Thank you. And sort of what I wanted to calculate is basically the fixation probability in a traveling wave. Right? So, we abstract this model here that we have sort of fitness and we have this Gaussian. And we now have, individuals that sit in different pieces, and each and every one of them could, in principle, be the ancestor of the future, right? And this ancestor of the future is the virus that we want to identify to predict what's going to be the future, right? So this is our population distribution, this envelope again, right? And these viruses, what can they do? They can mutate left and right, and I guess I have to increase font size again, right? So shout at me if it's too small. It's just so much easier to draw small things when you're so close. But yeah, you have a mutation distribution, right? Sort of a distribution of steps that you can take upwards or downwards in this fitness axis. And then what at least what's a convenient quantity to calculate is the probability of observing n individuals at time t, given we started at fitness x, and we'll have everything here sort of, fitness x, and this moves with the velocity v, right, just by Fish's fundamental theorem, right, is a variance, and the variance of selection on that variance propels us forward, right? And this is, so the probability that if we started a particular value of x at some time, what is the distribution of the number of copies at time t later? Right, and the way you want to calculate this, or at least one convenient way to calculate this quantity, sounds like a horrible beast to calculate, right? Because you have all this distribution here, and you mutate, and so forth, right? But it's actually not that bad. So let's look at this sort of n-axis here at sort of t equal to 0, and make a small time slice, sort of at t equal to delta t. And then we have, well, let's call this tau. And then we have here tau equal to t, right? And we start here at n equal to 1, right? And at some point later, we'll have probably a whole bunch of things that go extinct, and then maybe something like this, right? So that would be a distribution at some later time point. So how do you calculate this? We'll do this in what's sort of called a first step equation, right? So from this time point to that time point, there's multiple things that could happen. Either nothing happens, right? It's a small time interval. Maybe it doesn't mutate. Maybe it doesn't divide, replicate. It could replicate, in which case, or in this case, infect somebody else would be the replication aspect, right? Or it could, that person that was infected could become healthy, locked him or herself up at home, nobody got infected, then you go to 0, right? And that's game over. Right? So this is what we can write. Easily comprehend what happens in the short little time interval, and a mutation can happen too, right? So now we'll sort of write down this. Let me stick to the notes. P of n t, sort of given x, we write this as, so the probability of nothing happening would be, one, most of the time nothing happens, right? And it could mutate, it's delta t times mutation rate. Then there is the bursts, right, which our units are generation times. So it gives birth with that probability, and it dies with that probability, right? And this would now be all right. We write this first. So this is just the first term. There's more terms to come. So we go from here to here, either by nothing happening up to here, and then we have to cover a time interval t minus delta t, and our fitness now has decreased a little bit, right? Because that distribution in that delta t interval has actually moved forward, right? And fitness we'll measure relative to the mean, which is sort of the relevant quantity on all times, right? So this here is going from here to here, right? So we break down going from here to here into going from here to here and here to here. T minus delta t given x minus vt. V delta t, sorry. That's correct here. Right, the next thing that could happen is this des term, right? So we'll have delta t des, but des means the only thing that could happen in the end is actually n equal to 0, right? So there is a delta n equal to 0. This term does not contribute to any n other than 0. So we'll have, let's deal with the mutation term. So we have delta t and then all the different mutations that could happen. Right, the integral over this mu of s is supposed to be just this mu. Yeah, there really also needs to be sort of a v delta t here, but that it ends up being a second order term, so I'll just ignore it right away. And then there is the giving birth term, right? Which happens with rate delta t 1 plus x, right? And now, once we are two individuals here, there's many different ways I can give rise to some other n, right? And I'll sum over them. Yeah, so there's really all sorts of extra delta t terms here, but I'll ignore them because there's a delta t in front and everything else is going to be second order. And the blackboard won't be long enough if I... Right, so I can make n offspring by dividing and then have n prime offspring from one of the daughters and n minus n prime of the other, right? All right, so this can be rearranged a little bit, right? But you see where I'm going. You know, this is going to go over here, and then I expand this in delta t and x, right? So what you'll get from this is then sort of deep d dt. Now all the arguments are the same. Plus v d dx p, right? So this comes from here. That is going to be equal to minus mu plus 2 plus x p plus delta n equal to 0. And then what we'll do with this guy, you know, of course, this is a very general form. It could be all sorts of distributions here. Let's just assume s tends to be small compared to the relevant gradients in x. That's kind of the model I didn't present on Monday, but it's going to be fine. So we'll just expand this guy in S, right? So we can write this as P n t x plus s d dx p plus s squared over 2 d, right? And plus higher order terms. And in order for the higher order terms to be irrelevant, the distribution needs to be reasonably small. So now this no longer depends on s, right? So we can estimate here the first and the second moment of the mutation distribution, right? So let's just and this guy, of course, will sort of knock out this term, or just putting it back where it was. So we can get rid of this, and we'll have where am I? Yeah, plus sort of mu average s dp dx plus mu second moment dp dx squared, and we still have this n prime term here. All right, so that's already better. Any suggestions what we should do now? What's kind of annoying is this guy, right? So it's essentially it's a convolution, right? What do you do when you have a convolution? Fourier transform generating functions, something like that, right? That's exactly what we'll do. So we define sort of p hat, sort of lambda n p n x. Right? And when I drops the arguments, there's always going to be an x and a t, right? But we'll exchange the n for the lambda. All right, so this is linear up until here, right? So here, up until here, we basically just put hats on top. And that this convolution generates a square. Is that happy? Is everybody happy with that? Because you probably saw this from Luca 2 weeks ago. You did a branching process. Long time ago, 2 weeks ago. Yeah, so you'll get dp hat dt plus v dp hat dx. Oh yeah, so this is the one thing that sort of does something slightly. It just adds a 1, right? That's the one thing that's not a p. And yeah, so let's so mu delta s p hat prime plus sort of I call this now. So this mu s squared second moment I just call d. Let me just quickly make sure. I think I call the entire thing d. Right, so you end up with something like this. Then it's actually quite convenient to define that guy, because the formula will simplify a lot, because there's all sorts of ones that then disappear. And the result is going to be this d phi dt minus v dx d. Did I screw up something? Oh, yes. So that guy is here twice. Then the question is, is the sign still correct? No, the sign is then, this one needs to go to very quickly. Yeah, that should be correct. D. Is it's just it's basically the diffusion constant in fitness space, right? And this here is the advection term in fitness space, right? Okay, p prime. I was inconsistent, yeah. Right, and this here is going to be sigma squared, the variance of the distribution. Yes.
So, when these things are evolving, so won't the environment play some role? Like the mutation distribution, does it depend on the environment or something?
Yeah, but I think at least in this fluke case, of course there is not this constant mutation distribution, right? But there is an argument that can be made that over the past 30 years, it's just been sort of running in a treadmill, right? The humans change the immunity profile, and the flu changes the immunity profile. Of course, that distribution will change a little bit here and there. But the overall statistical properties of it seem to be not too different now from what they used to be. All right, so this is an equation for the generating function, and there's a few simple things that you can do, right? And the easiest is probably just calculate the mean of n at some time, right? So you get the mean from this guy, right, by differentiating with respect to lambda and sending it to 0. And since differentiating that. So where am I? Differentiating that entire thing with respect to lambda. Lambda actually is sort of implicit, is a variable of all of these guys, right? But this is just all linear. And this guy is quadratic. So you differentiate that with respect to lambda, you get this. But then you evaluate this at lambda equal to 1. And lambda equal to 1, this guy vanishes. Which I didn't specify the initial condition of all of this, right? So my bad. So sorry. Somebody is paying attention, that's good. Right, the initial condition of all of this, p hat lambda 0 of x is just lambda, right? N equal to 1. Probability 1, right? So we have to evaluate this at lambda equal to 1, and that means phi is initially 0, and if phi is initially 0, it actually never gets out of there. Right? Identical 0 is a solution of that equation. So this is why this term just drops, and we are left with a linear equation for the mean, and the mean n is then just x times the mean n minus sigma squared sort of d mean dx plus d which has a very simple solution, is e to the x t minus v t sigma squared t squared over 2 plus dt cubed over 3. Which is to say, if we do sort of log n here, we'll have, of course, there's those, if you are initially at fitness below the average, right, you're not going to go anywhere, right? Even though this term nominally rescues you at some point, but you have to go through a lot of fractional individuals to get there. So this is something where extremely rare events sort of mess up the average. If x is positive initially, right? So you will grow, but then the mean catches up, and you'll start to shrink again. And maybe at some point you'll come back, but that is again non-physical, right? And this corresponds here to being somewhere here and growing into kind of a bubble and then contracting again, right? And you're contracting because the mean is kind of rushing, so the population is rushing past you. If you have a large enough population, then you'll actually go through an inflection point, and sort of the different extrema of this will collapse. And this is kind of when you have to then connect to the coalescence theory. So you can do all these calculations as a branching process, as long as you're not materially affecting the mean of the distribution, right? Because the mean we have assumed here to be externally specified. Once that happens, you're basically grown as big as the entire population, and you have to start doing a different calculation. But that actually ends up being exactly the threshold where sort of the coalescence then happens. So this is sort of one way of looking at it. The other one, one. Oh, this is different x's. So this is some xc, right? This is a small x. And this xc actually ends up being, that's related to v, right? And that ends up being the place here where the tip of the distribution is, right? And that xc, that is actually, xc ends up being sigma to the 4th over 4d. And it corresponds to a time. You reach this inflection point after a time, which is, why doesn't it say here? It really should. Sigma squared over 2d. And that ends up being the coalescence time. So now, the reason we really did this, right, we wanted to understand something about fixation and being the ancestor of the future. So far, I've just told you what the mean is, and we probably could have found an easier way to calculate that. To get the fixation probability is essentially the probability of all these terms other than term 0, right? So like being not extinct, so non-extinction probability, survival probability, which is, if you evaluate this at lambda equal to 0, right, you get the extinction probability. 1 minus that is the survival probability. So phi evaluated at lambda equal to 0 is the survival probability. So that means if you now look at this equation, with initial condition lambda equal to 0, means here initial condition 1. Right? So we are looking at, I don't mess this up, something that x coordinate here. This is sort of x equal to 0. And let's sort of have 1 here. So at t equal to 0, our phi is like this, right? So it's phi is large. And initially, x is typically, the growth rate in one single generation. So the axis fitness compared to the mean in a single generation is typically small. Like fluke A is maybe 10-20% or something like that. And that would be pretty big. We initially also don't have any derivatives, right? So all these terms are initially 0. So the dominant balance initially is going to be between this 1 phi squared and d phi dt. So phi dot initially is going to be minus phi squared. Suggestions? phi is initially going to be something like 1 over time, right? Meaning it sort of will come down rapidly and indiscriminately like this. At some point, however, it becomes, so 1 over t becomes comparable to x. Right? So once 1 over t becomes comparable to the selective advantage, meaning selected advantage times time is of order 1, that's when selection starts to matter, right? And then very rapidly the dominant balance here shifts between and that to this and that. Right? Then we are dealing with the new dominant balance between x, phi, roughly phi squared. It's even simpler, right? So phi is proportional to x then. And that means, so this is actually starting to peel off. And you probably start to recognize one of the graphs from the review papers, right? So this is, so it starts to peel off like this. But then, of course, we start to introduce here a derivative and a curvature in the crossover. And while it's sort of continuously going down here, it's becoming smaller and smaller and smaller over here, is 1 over time, right? And in this region, then, you can start sort of solving this without this bit, because the phi squared, once it's very small, the phi squared doesn't really matter anymore. And you might remember that this equation is very similar to the area equation that you have from WKB approximations and that sort of thing. So you get in an area solution times an integrating factor over here, then a t to infinity solution that looks roughly like this. And this point here, where it sort of joins this x line, right, that is roughly at xc and coincides with the tip of this traveling wave. And the interpretation is that if you are starting out as the fittest or above the fittest in the population, the probability that you take over everything is purely determined by whether you survive just initial stochastic drift, right? And initial stochastic drift, the probability of fixation of just one single mutation, you might remember, was proportional to S, which is the same thing as X here, right? And all the way out here, if you start out with a fitness at this level, you're golden. You only need to divide a few times and then run off, right? But once you start to compete with other people, so once your fitness is below this XC and you're starting somewhere here, right, then the chance of succeeding strongly depends, first of all, where you started, and sort of various other parameters like how you jump around and so forth. And there's the region here in the top where there is an appreciable chance of being a common ancestor of the population, and below it's exponentially rare. And where this XC is and how wide this region is, that depends on the population size essentially, but only in a logarithmic fashion. All right, I have another 10-15 minutes. And in this remaining time, what I wanted to do is go back to the slides and show how, at least ideas building on this. This is not the whole story, right? This only tells you fixation. It doesn't really sort of take it back to the tree, but that would be sort of a, more convoluted, and I'd need both more preparation on my part and time of yours to do a satisfactory job on that. So I'll just do a somewhat more hand-waving explanation. But it's good to have this as a background, as a kind of calculation that you do in these models, or using these models to link them back to data and inference. So is this projector coming back to life? Yeah, I think something's happening. Oh, Panasonic. Well, XC is the typical position where the fittest individual in the population sits in the fitness space, right? XC is kind of the pioneers. All right, here we are. So the problem is, nobody tells us, well, green on green doesn't really work, right? Nobody tells us where our individuals are in this fitness space, right? So we need to, all we get are the sequences. So we need to find a way, work our way back from the sequences to fitness. And last week, I sort of very briefly told you about this Bolthausen-Slittman coalescent process, which comes out of these traveling waves, right? Due to the fact that the things that sit in the top of the fitness distribution, that they rapidly increase in numbers, according to this average formula, and then sort of generate in these bursts of offspring that they leave in the tree, right? And the defining feature of these trees are that they have these, what's called a multiple merging event. So going backwards in time, many lineages come together almost immediately. Forward in time, that corresponds to an individual that here in the tip kind of hopped forward, kind of past XC, if you wish, right? And then sort of rapidly grew into a big clone from which a large fraction of the population descended. So in a way, these bursts that you observe in the tree, those are kind of echoes of exceptionally fit individuals that lived at some point in the past, right? And in the context of flu, right, what we'd like to do, we have a population sampled in a particular year. We'd like to anticipate the future, meaning we'd like to identify a sequence that's as close as possible to that common ancestor of the future population, right? And that's why, and where this individual sits in this background fitness distribution, right, that's a calculation that we just did.
So this phylogenetic tree, instead of using the whole genome and doing the alignment and reconstructing the tree, are there certain sites that actually recapture most of the tree?
No, the tree is inherently a property that is reconstructed from as many sites as you wish. And the sites that change most rapidly, meaning those that are actually under selection, those you don't usually want to use for tree reconstruction because you get a lot of repeated mutation in those. But these are not necessarily informative of history. So the tree itself is best reconstructed from synonymous mutations, and as many of them as possible.
Thank
you. So you'd like to have a way to identify this guy, and we sort of have an understanding of where that guy sits in the fitness distribution, but we have an intuition, right, that things like this in the tree will correspond to high fitness individuals, but we haven't formalized that yet, and I'm not going to take you through that, but I'll give you some sense, right? So we have the tree, right? This is a sample of individuals, and ideally we'd like to attach a fitness score to every single node in the tree. And what sort of Boris Schreiman, Colin Russell, and myself, we sort of wrote down, doing some approximations in that we assume that different parts of the tree are at least transiently independent, that we can write down a posterior distribution of fitness, X being fitness, conditional on the tree topology and branch length. And due to this tree likeness of this, you can write this as a product of sort of propagators along the branches. So the tree, there's no loops in the tree, right? That makes many calculations a lot easier. And each of these branches is associated with what we sort of call a propagator that quantifies how likely it is that the top end of that branch had a certain fitness, it had a certain length, and the bottom end of that branch had a certain fitness, given that there is sampled offspring. Given that there are offspring in our sample sequences, right? And that the branch is not branching again, right? So it's conditional on observing a bit of the tree that is unbranched but leaves offspring. So that is what this object is. There's lower end fitness, upper end fitness, and the 2 times, right? And it's important to condition on both having offspring, because otherwise it wouldn't be part of your tree, and on not branching, no branching happening here that has offspring. Okay, otherwise you have multiple branch propagators, right? In a way, you have for every vertex, you have sort of a vertex factor. For every branch, you have a branch propagator, right? And you write down a distribution like this. And so this fixation distribution is actually an elementary, is a central part to it, because that tells you the probability of being at a certain fitness X and ending up in the sample. And you don't use lambda equal to equal to 0 in this case, but sort of like some epsilon, which is your sampling fraction. And these you'll have to evaluate numerically in the end, because the nonlinear equation a nonlinear equation is behind the board. You can't actually solve, but you can evaluate numerically, and then you tabulate all these things and you multiply them together. And you can trace and calculate marginal fitness values for each node. Because it's a tree, it's easy to marginalize. Okay? So what you'll end up getting is sort of the background fitness distribution, and then for a particular node, you get sort of a marginal fitness distribution. And in this case, this node here would be a pretty good candidate, right? Because it sort of is a burst that happened recently in the tree, while this lineage here probably doesn't go anywhere, right? It's kind of some left over. Yeah. The black dots here are observed ones, and the gray ones are internal ones. Yeah. Well, so what we predict is we try to identify the sequence of highest fitness. And the sequence we know, right? So the amino acids that come with the sequence, the amino acids are not the mystery. The mystery is the ranking. Yeah. Well, we do the internal nodes as well, but in Flu, pretty much every internal node is also represented as an external node. So it's yes. Well, they branch all the time, right? But it's conditional on branching and not ending up in the sample. And that's if you only conditioned on having offspring, the most likely thing would always be as fit as possible, right? So that is so the condition on not branching basically pushes your fitness into a defined range, right? Because if you're super fit, it's unlikely that there is one long stretch of tree that doesn't leave any offspring, right? It should be just overgrowing everything and producing lots of offspring here that you actually sample. Right? So there's sort of two opposing forces, and it's basically 1 minus phi, which then sort of comes in as an opposing force. That thing ends up being the potential that pushes it to lower fitness, and this guy pushes it to higher fitness, right? Right. So what we are hoping to do is find the black dot, which is as close as possible to the common ancestor of the future. Right? Because that would be the closest representative of the future population. Right? This one here would be you have distances on trees are ultrametric, right? So in the distance between this and that would be going all the way up here and going all the way down here, right? The distance between this and the future is just taking this little detour. Right, so this would be the closest representative of the future. Yes, that is a valid way of doing it too, and sort of my friends and colleagues, Marta and Michael, have done that, right? So it's this is sort of a very agnostic way of looking at it, right? Sort of is it just in the pattern of how things spread? Is there something that we can say about who's likely to go forward, and not understanding the molecular, biophysical, antigenic, immunological, and whatnot properties of influenza? That is, which I don't. It might be a useful way to go, but let's bear with me. All right, so in essence, the first thing that we did is sort of test it in simulations, right? You can simulate sequences under your model, and then just write down ACGT sequences from your computer, which mean absolutely nothing, other than that they have a history and they have been evolving in your model. You reconstruct a phylogeny, you throw this algorithm at it, and ask, can you predict I thought there was another slide here. Okay, can you predict fitness? Yes, you can predict fitness, but not terribly well. You can predict fitness with explain 30% of the variance. But there's a very clear sort of robust correlation in predicted fitness and true fitness. What's shown here is the degree to which you are able to predict the sequence of the future. And what this plot does, say you made this prediction here. It's a bad prediction in this case, right? But what this graph here shows is the distance, the nucleotide distance, or the tree distance of this dot to the average of the future, normalized to the average between these, right? That's the dashed line here. And what you already see is that the majority of these points are below the dashed line, meaning we're making a prediction that's at least better than randomly picking, right? Because that's where we would be in that case. And on the x-axis, we compare it to the post hoc optimal choice, right? So if you're here on the diagonal, that means you've actually picked a representative that is close to optimal. And from these simulations, and there's many of them, right? You roughly get a picture where sort of 30-40% you actually picked the optimal clade. Then there's sort of 30-40% that are kind of middling, and then there's a few cases where you're random or worse. But it's certainly much better than randomly picking a sequence. If you now do this with flu, and that's actually that's just a little animation where you have the red dot being sort of a prediction, and the gray dot being the vaccine. The red dot's here, and you see that the red dot basically always moves like half a year to a year before the vaccine. Now the vaccine ends up being super audacious. I think they're up right here. So that was the one time where they actually made a huge, big, and prescient move. But now, the vaccine's going to, and then that's going to be. But I have a more quantitative picture of this as well. So here you see, again, that measure of, prediction relative to average, and the lower level here is always the optimal. And in a few years, right, you do pretty optimal. In a few years, you're kind of randomish, and then a few years in between, so somewhat consistent with what we did in got from the simulations. And sort of what's remarkable about this is, that works without any influencer-specific input, right? And in fact, what's shown here is not this complicated model that I've just taken you through, but what you can do, you can analyze a few asymptotics of this model and show that it, in many cases, just reduces to some local tree volume score, which we call the local branching index. And if we just go back to that graph, you basically, for every part of the tree, you ask, in a particular distance around that node, how much length of tree do you observe? That's a measure of how many lineages go out, how rapidly is this branching. And sort of all of this fitness prediction basically boils down to finding sort of little parts of the tree which look very bushy, where many lineages of explosively suddenly come out of one. Which is exactly what we started out with as an intuition, only that this intuition we really only developed post hoc. So I told you. Yeah, so I'm not going to tell you anything about that. This was just, I didn't quite know, and then I was I do it or not, but I didn't take it out. But I can tell you, just a few minutes, show you our sort of other efforts to take this kind of real-time tracking ideas to different diseases. And I think I have that sort of preloaded here. So it's again a collaboration with Trevor Bedford in Seattle. And what actually mostly he by now has set up is such sites for Zika virus, Ebola virus. We're starting to look at dengue and the different serotypes of it. There's avian influenza, mumps and measles, because there have been recent mumps and measles outbreaks in various parts of Canada, the US, and the UK, which they want to track. And sort of what I have here is sort of the Zika page, which is sort of currently probably the best developed. So what you have on the left, as before, is sort of phylogenetic tree. So it lives here on a time axis. In 2014, that virus sort of suddenly showed up in French Polynesia, which here on the map are these bits, Tahiti. And then from there, in probably late 2013, got transmitted to Brazil, and in Brazil sort of rapidly spread through the entire subcontinent. Until about now, 2/3 of the entire population is seropositive for Zika. So it really infected, mostly subclinical and unnoticed, a substantial fraction of the South American population. And this only worked because the population was completely naive regarding this virus. It has been circulating in Southeast Asia and Africa for a long time. But like a firestorm, burned through a susceptible population in South America and has been then spreading into Mesoamerica and North America, Florida a little bit, right? And sort of what this tool here, for example, lets you look at, that was not supposed to happen. So you have the American viruses in red here. So these are all these guys. Right? These are mostly sampled in Florida, and they're embedded in viruses that got sampled in the Dominican Republic or other sort of Caribbean islands, which sort of tells you that probably most likely these viruses traveled from the Caribbean to Florida by tourists that sort of came back from spring break or from cruise ships, and then sort of once they arrived in Florida, so the dot here, the US dot is a little misleading. It should really be here in Miami. But it has then sort of led to local transmission chains of Zika, which in some of these sequences are actually isolated from mosquitoes. And this is a result of multiple separate introductions, right? So this clade here is one separate introduction, that is another one, and then there have been a few more isolated ones, because they are nested in the diversity of the Dominican Republic. And it's really not all that hard to make these things, right? To polish them and make them, look very pretty. That's a lot of work, but it's pretty straightforward to set such things up these days, because there's all sorts of web technologies that Facebook and Friend developed that you can use to make a very data-driven. D3 is actually the engine behind much of this. D3 is sort of data-driven documents by Mike Bostock, who spent, there's a fabulous toolkit to build things like that. And it's not that hard. You just need to be persistent and imaginative. And the data is there too, right? So just don't be inhibited. But it just needs to start, and then you get somewhere.
To add malaria.
We've been thinking about malaria. Malaria is sort of a much more complicated beast because it has a huge genome and it recombines. So what we are starting with now, going this direction, is TB, tuberculosis, because that is easier than malaria. But I think once we come to grips with tuberculosis, we could potentially look at malaria too. So there's no sense in which malaria will have a tree. But there's other interesting things that you can do, right? So you have trees in particular regions, so you'd like to understand how artemisinin resistance, for example, spreads, and that would have a tree. But yeah, so you would need sort of a different underlying processing toolbox. All right, it should probably come to an end, and I'm kind of at the end. Is just sort of how this works. Yeah, so I hope I gave you some rough sense of how sort of applied evolutionary biology can be used to track and improve our response to infectious disease, and how even we got into this from a completely academic angle, right? We wanted to understand these models and these trees, right? And there was no link to anything applied at the time. So the most applied thing we were thinking about was sort of Lenski experiment kind of thing, right? And then there was sort of an intellectual challenge. So yeah, is there information in the sequences about fitness, right? And we worked on this for 1, 2, 3 years, and then suddenly there was something that made sense, and we needed data to test. And then influenza was the most obvious thing to do, right? And before you know, you end up talking to people at the CDC and the WHO about what to do. So it's I think it's a very, very nice tale of how completely curiosity-driven, abstract, blackboard, pencil, paper kind of thing. Before you know it, you are talking to public health people, and that's not always enjoyable. Yeah, and actually building these real-time analysis tools has been a lot of fun, but that's kind of the thing I like to do. All right, so I should thank these people. Sort of Boris was, the driving force behind the fitness inference kind of thing. Oscar did a lot of work with me on the coalescence. Trevor and Colin are sort of our, those people that know more about flu than we do, and a lot more than we used to know. And then there is sort of the team behind NextStrain, which involves 2 of my graduate students who are just about to defend, and Colin McGill, who's a React developer, and James Hatfield and Sydney, who are in Trevor's lab. Sort of Sydney mostly does the dengue work, and James Hatfield does sort of nuts and bolts of the visualization. And with that, thank you for your attention. Happy to take questions. I'll be around for another 6 hours. And if you have questions or want to bug me in various ways, feel free.
So if I understand, correctly, it's your fitness prediction is essentially predicting which strain to choose to make the next vaccine.
Exactly.
Right. And
yes, that's correct.
Decide which is the next coming up strain. Which will be the
but that is a related question, right? So we are predicting what is the strain that's most likely going to take over, and that's the strain that you probably want to use for the vaccine. Only that for the vaccine, of course, you have all sorts of other constraints that you also need to fulfill in that it's a virus that sort of behaves well in production and so forth. And it needs to be sufficiently characterized by the time you go into the meeting, because you can't sort of go for a virus that is basically an uncharacterized wild card, right? So these influenza researchers need to do a lot of work before you can decide this is the one we use. So
So, quick clarification. So you predict which is the next strain by measuring proximity, by measuring which one is closest to what is the most fit strain, right?
Yeah, well, among the sample of viruses that we have, we're trying to find the most fit.
Right.
It's essentially sort of saying that you'd like to calculate the derivative of the frequency of a particular clade.
Right.
And the intellectual challenge at the time was, of course, to do it without temporal data. For flu, we do have a lot of temporal data, right? We don't need to be as restrictive as this. And we do now, of course, at sort of how these frequencies of different mutations and clades change from week to week. And that sort of, that should in many ways be better, right? than sort of, but in fact, you don't get much out of it, because this fitness inference and these local branching index, it tends to just average growth and sort of little fluctuations over just about the right period to give you some robust thing.
The black dots on a slide, that you were trying to predict which.
That's like 100 slides away.
That's okay. Yeah. So you're trying to predict which is the closest to the possible future.
Yeah. So we're trying to find this guy, right?
Right. Okay. And okay, just wanted to clarify that. And a quick computational question, maybe slightly off track, but your next strain work, have you thought about potentially partnering with Google? Because Google tracks web searches of the flu. So it would be interesting if you could integrate people searching for the flu and...
Yeah, but what Google Flu Trends gives you is basically the same kind of information that I showed you in the first couple of graphs, which is sort of flu incidence, right? So that's kind of the envelope of the population size. And people are not going to search for mutations sort of 198K, right? So we are sort of trying to look at the dynamics within the population, which is, and I think it's a worthwhile thing to consider. So how can we use incidence trajectories in different geographic regions to make this sort of analysis better and more robust, right? Because right now, these different frequency changes and so forth that we see are sort of a mixture of things that come from different geographic regions, and to the extent that different regions have sort of slightly out of sync flu population dynamics, we might get all sorts of, aberrant signals from sort of fluctuating envelopes of the populations. So there is certainly a lot more that one can do. The main challenge that we have is that we have only 20 years to test those things on. And the moment you try to do something more complicated, you introduce 1 or 2 or 3 additional parameters, which you just have no way of pinning down and make it robust for against overfitting.
You actually sort of answered it, right? Having more samples only sort of changes things by log n, right? But on the other hand, in this picture, right, all the black dots, though the black dot on the right looks like in the tail, but with very low sample, it's actually right in the middle, right? At least that's how I'm imagining. Maybe I'm imagining wrong. So in that sense, it feels like more sampling should help. Is there a scaling, some version of thinking about it?
But sampling into the tail requires exponentially more.
No, precisely. No, precisely. So what I do, is there sort of a trade-off? Is there a way of thinking? So I understand that, right? But so in some ways your black dot, you're actually getting somewhere near within the variance, right? But what you want is basically some guy outside.
Yeah, so, flu is somewhat more forgiving in that we don't actually have, we have a bunch of major clades, right? So the distribution is not that smooth. And every major variant you'll get, and then you sort of you go to the most to some recent common ancestor, and that gets you into the tail. More samples do help when you have geographic structure, right? Yeah. Because then, so that's where you actually gain information, right? But just more samples from New York is not going to help us much. Yeah, I think he was first.
Right. So I think the part of the vaccines is out of the equation so far. Is it like, have you studied the effect of introducing the vaccine in the tree?
Yeah, that question comes up every time. So right now it's not too much of an issue. So who's vaccinated against flu? See? So does that answer your question? No, we have even the data on the efficacy of the vaccine in a given year is very sketchy. So you're asking too much at present. But you don't have the question.
So as you said that there is some amount of fuzziness in determining which is the fittest one. But could you, so the other approach which you said, could you combine the two approaches, the biophysical approach, to maybe better understand?
Yeah, so that's obviously something that, so we would like to combine the different approaches like Michael's and Marta's model and ours and sort of, fold in sort of many different things. The problem that we have right now is that each of these things individually already works pretty okay. Putting them together makes it a little bit better, but you also have more parameters to trade off these different components. So we are not confident that it actually makes it a better predictor, makes it a better postdictor. A postdictor, right? It's a better fit to the past, but it's not obvious that it's a better predictor. Yeah.
Right. So, I mean, my feeling is that by introducing the vaccine, you're changing the fitness landscape. That's the thing you want to do, right? As a WHO. So, are you saying that? What are you saying? Sorry.
Well, so in the way it's working now, right? That the vaccine is basically a virus from the past. It's sort of making the immune response of humans stronger, kind of pushing the virus forward a little bit more. At the same time, however, reducing virus transmission and sort of taking out evolutionary potential in principle. If you were to actually anticipate and kind of, or maybe even sort of jump ahead and sort of produce a virus that is sort of in the future, then you would have a chance of steering the population somewhere else, but nobody has tried that yet. At present, in vaccine coverage, in particular in the most populous countries on the planet, is low enough that we don't think that this is a substantial driving force of influenza evolution. And so that was my point, right? So, places like here, Southeast Asia, East Asia, those are considered to be not exactly the source, but kind of the region where most of the influenza evolution and dispersal